This is a production of Cornell University.
Thank you, Alina and Mike, for inviting me. I really appreciate the opportunity. Oh, sorry, can you hear me with this mic? It's okay? Great, okay. Yeah, so as Alina said, I'm sort of known as a root imaging and root analysis person, but really we're interested in how the plant functions as a whole and how shoots and roots are integrated the way a plant actually works. And so this is actually data from the PET imaging system, my colleague Yanshan Tai at the WashU Medical School. So you're actually watching carbon being fixed in the leaves and moving down into the roots in real time using PET. And I'll tell you a little bit more about how this works later in the talk. But again, as I think we all understand, if we want to understand the plant as an integrated system, it's usually the roots where we stop, right? These are the bottlenecks. So this is a classic image of two grass roots interacting with one another. So there's just one little shoot up here, quite an extensive root system. So this is one scale, the architecture of the root system, that we can look at roots. But similarly, there's at each of these many thousands of root tips, there's actually a microenvironment here, the rhizosphere. So this is the inside of a barley root. You can see the root hairs here, the soil particles. This is where the microbiome is happening. So there's a lot of dynamic activity across a lot of scales. So suffice it to say, roots are an incredible challenge, not just to see, but in their complexity at many levels. And so from an applied standpoint, we often think about the concept of idiotype breeding, which was developed by C.M. Donald in the 60s. But an idiotype is basically a model plant trait for a particular environment or agronomic practice. And it was a big deal in the Green Revolution. And the idea is to breed for certain plant characters or plant traits rather than simply for yield or for reduced defects in yield. But as you might imagine, Donald didn't have a lot to say about what the below ground idiotypes are. Nonetheless, all the water, all the nutrients, and quite a bit of interactions with the environment come through the roots and through the soil. And so this is a really key area that we focus on in the lab. So this is how those images that you saw, this is from John Weaver's papers, really pioneered this sort of archaeological approach to seeing and understanding root systems in the soil. This is literally what they were doing through the early 1900s and through the 50s. And as you can imagine, the throughput was quite low. Unfortunately, 85 years later, we're literally, this is still state of the art. And this is not much hyperbole here. And so of the different ways that we can actually look at roots in the field, each one has a very limited perspective. It's a very sparse idea of the whole root system. So there is a phenotypic reality. It's three-dimensional. It's actually growing. So it's a four-dimensional reality. And each of these root tips is growing along in its microenvironment. It adds up to a total architecture. These are the methods we can use today in the field. Excavating, soil cores, mini rhizotrons, which is a clear tube with a camera where you can just see the roots growing along the tube, or root crown excavations, aka shovelomics. And so just the idea here is that there's, of course, useful information we can get, but it's only a little sliver of the real picture of phenotypic reality. And this is sort of a theme in my work. So we're not only interested in seeing the root system, but sort of making sense of the complexity as well and tying different forms of data together. So ultimately, my group is interested in understanding the genetic basis of root growth and root environment interactions. And we very much take a kitchen sink approach. And there's two general axes here. One is just experiments we can control, the information content, and on the other end, what's actually relative. Is it field soil? Is it actually relative to agriculture? And so there's a number of different methods, and I'll touch on most of these today. But we take the kitchen sink and we throw it at what I like to call high resolution germplasm. And the resolution is the resolution to identify genes that are controlling these traits. And so I'll tell you a little bit about the Illinois long-term selection experiment in maize and how we're using these tools to identify genes in a little bit. First, I'll start with an imaging system for looking at roots that was actually developed here in Leon Kochian's group at Cornell. This is optical projection tomography, and there's many of Leon's former lab here, at least. Randy Clark was the one, and John Schaff, who really pioneered this. It's nothing more than growing plants in a big glass jar with fertilizer and gel and spinning it around on a turntable. So here we have a camera and an illuminator, and we're basically capturing silhouette images as we're rotating. So we capture three-dimensional information about the roots. And from that, we can actually make a three-dimensional model. And once we have this model in hand, we have to figure out what to measure about it. So we literally have now a phenotypic reality, but how do we capture all the variation here? Well, typically we start with the things that we could measure maybe by hand or with a ruler previously, and then we get into fancy stuff like shape analysis, like topological features or how the roots curve over time, things like this. So there's a lot of information we can get, but I'd like you to keep in mind that these are really, they're discrete traits and they're ad hoc. So they're basically our human idea of what is important for the root system, and to some extent this may be limiting our ability to understand roots and identify genes. And I'll give you a little bit of an example. So I'm skipping ahead, but we use this method to map the root architecture of a rice mapping population. So it's an upland and a lowland, a rice, Balanasi, Sena, recombinant inbred lines. So Mike explained QTLs and RILs earlier today. So basically, what I'm showing you here is just a few different chromosomes where we found QTLs. And hopefully you can kind of see the shaded areas, the color map. This is where we found lots of QTLs, these univariate QTLs. So again, we may have found one for bushiness or one for root system volume, or maybe even together in the same QTL here, okay? But we're wondering, well, which QTL should we actually pursue? Which of these traits actually matters most? And this is where we're working with Tom Mitchell-Olds. I started to get a perspective, a multivariate perspective about phenotype. So what Tom did is he took this whole list of traits, everything we could measure, and Jill Anderson and Ray in his lab, and just used a MANOVA, and basically used a multivariate MANOVA as the trait. So a similar thing would be to use a principal component and map on a principal component. And the point of this slide is just to show you, we found 5 multivariate QTLs. The ones in red here, and this is the effect size, so they're pretty large. In red here, these are ones that we found with the univariate traits previously. So we already had evidence that there was some genes controlling the trait of interest. But in green here, there was no evidence of univariate QTL, meaning we would not have found this using these discrete and ad hoc traits. And I think this is a very important concept in phenotyping in general. And I think all the speakers this morning have really hit on the fact that there's lots of information and that together they tell the picture and maybe not alone. So a really beautiful thing about this gel, albeit an artificial system, of course, is it's non-destructive, and so we can actually look at the 4th dimension, time. So when we think about plant environment interactions or dynamics or processes, all of these words involve time. They imply time, and so to not look at them over time is severely limiting. So here we're looking at 2 maize inbred lines, and we can see that there's very different root architectures, but what we would like to know is how the growth of each of these individual roots, how it goes from one root to many hundreds of roots over the course of 7 days, and how that process is. So how are they different as a function of time in their growth processes rather than just a static time point? So this is an automated imaging system we have in the lab. It's just every 4 hours or whatever we decide to program it at, it runs day and night, and so we get these really nice time-resolved data sets. We've also developed a software, it's Dynamic Roots, published a couple of years ago, takes these 3D models and allows us to actually measure the growth of each individual root in the context of the whole root system over time. So we can track individual roots, we can look at a very highly resolved level temporally and spatially. So this is work of Ni Jiang, a postdoc in the lab. So again, just these 2 different maize genotypes. Here we're looking at root length, and so if you just focus here, it's shown as in a density plot. So as a function of the total, so basically in blue here, these are relatively shorter roots compared to the relatively longer roots, and that's the same here for Missouri 17. And so this is just to show you that at the beginning, B73 has relatively longer roots, and that continues for some period of time, but at some point there's really a flush of lateral roots, and then the architecture continues with many smaller roots rather than many longer roots, whereas Missouri 17 over time invests more in the roots that it actually has and grows these lateral roots out rather than making more new ones, if that makes sense. So this is really, could be thought of as a foraging strategy. It's definitely a growth characteristic that has implications for how the plant is able to forage for nutrients and resources in the soil. We can also see that, clearly, depending on where you looked at your trait of interest, you would get different answers. So if you looked here, it tells you one thing. Here, they're very similar, and here you get a different answer. So similarly, this is just the number of lateral roots. So basically what I showed you before, but you can see they're very similar numbers until some point at which B73 ramps up and moves across. So how do we actually capture all of this information in a more comprehensive way? So really in a function rather than discrete values. And so we've started modeling these growth curves. And so taking these patterns in the data and converting them into a few variables that we can then use to probe the behavior and use as traits for the genetics. So this is, again, a very basic function. Most biological things start growing and they grow kind of like an S, and that is modeled well by a logistic function. And so you don't need to read all this. I'm just obligatorily showing you the equation here. But the point is that there's just a few variables that we can use to capture the data, so it's probably better looked at here. Again, this is total root length, B73 Missouri 17. So at the final time point, this is after about a week of growth, B73 has a more total root length, but it's pretty similar, okay? And this value is captured by one of the parameters, the carrying capacity, alpha 1. And so we can see that, here, where the alpha 1, it's a little bit bigger for B73, but not statistically different. Then there's another trait, the inflection point, which is where the growth rate, actually you can see it here better. So the growth rate is speeding up for B73, and then it starts to slow down. So the inflection point would be right in the middle, and it would be somewhere on this curve. Missouri 17 ramps up a little slower, but then it ramps down a little slower as well. And that's captured by this value. The slope of the line is captured by these other two values. And so essentially we're distilling these growth patterns into variables that we can then use to differentiate the genotypes or eventually to map genes. Okay, so switching things up a little, this is what we can do with very high resolution data where we can see and know the whole root system. Of course, out in the field, which is where we really want to know how roots grow and behave, it's a lot different. So again, this is our field of corn. That's a foot with a shovel. That's a tank where we soak the roots. This is our mobile field lab. So it's got air conditioning. It's humane. We drive it around from field to field, pile out of the back, and just dig lots and lots of roots. But what you're looking at here, instead of measuring this by hand, this is actually a light, and there's a camera inside of it. Maybe you can see it here. It's a little dark. But we take formal portraits of the roots, and we actually use image analysis methods to measure the roots rather than to measure them by hand. And so for this, we use a software called DIRT. And DIRT is now on iPlan. It's free, so no excuse for anyone not to look at the roots anymore. Just wash them off, take a formal portrait, and there's a lot of information we can capture then from these two-dimensional images. And so I'm going to skip over this quickly, actually, but there's a plant genome project where we're doing a lot of this work. And this is, at the core of it, we're studying the Illinois high and low protein lines. So these lines have been bred since 1898 for either high or low seed protein content. All this protein is storing nitrogen. So these are really good at taking up nitrogen, and these aren't. So to the extent that root architecture should be conditioned by, or nitrogen uptake should be conditioned by root architecture, we should see the difference and so in the roots. And so for high and low protein, they may look different to you. There's something different about them, but what is it? Well, DIRT kind of gives us a lot of information about what it could be. And so if you cross these two things together, this is just phenotypic variation in the real line. So variation is good if you're trying to map traits. And we've now used this to, in a GWAS analysis, to identify candidate or SNPs that are near candidate genes that we think may be controlling root architecture that has some bearing on nitrogen uptake. So this is the work of a postdoc in the lab, Zhengbin Liu. Here's an example on chromosome 10 where we've drilled down to a candidate gene that we think is controlling root architecture. And we have a number of these genes, so the traits really don't matter. This one is for root system width, but again, these traits are just capturing some variation in the root phenotype. They're not really meant to be taken literally, is my perspective. So I'm showing you here the sorghum ortholog identity because we actually found mutation in an existing library in sorghum, so we think it's doing a very similar thing in sorghum. And there's our candidate gene, there's the wild type, and we can see clear root phenotypes here. So we think we're getting close to identifying genes. And this is important because we know almost nothing about the quantitative genetics that control root architecture. So another part of this project, and the NSF partially funded, and the other half of the money came from Valent Biosciences, who we're working with on root rhizosphere interactions, mycorrhizal fungi, and rhizobia and nodulation. This is an industrial scale X-ray imaging system, the X5000. So you'll find this in factory floors and automobile and aerospace industry. And so it's meant for big, heavy things that are dirty. And that's kind of what plants in pots are as well. So very talented research scientist, Keith Duncan, running this. This is what it looks like on the inside. So I've got a little schematic of how it works, but that's the source, that's the detector, that's a sample, and this thing can spin around, kind of like the optical system. But you can also see a lot of mechanics, so we can do very large samples. So again, quickly how it works, there's a source and a detector, and this sits on a very something with light density, and as we shoot X-rays through it, we get a contrast image. So the denser the material, the more it absorbs the X-ray, and that generates our difference in contrast. This is a pot with a cassava root system. It doesn't look like much, but there's more magic that happens. So we basically collect lots of these as we spin around, and then we can develop a three-dimensional reconstruction, and I'll show you a whole litany of these in a little while. But as other features of the system, after we've spun around, we can actually, because of the mechanics in here, we can actually move the source and detector up and scan across the whole plant. And obviously this comes at a cost of data and time, but we can do it about 4 feet. And we can also do what's called a mosaics, which is a tiling scan. We can scan across, and so we can do very wide things. So we have about a 2 by 2 by 4 foot imaging volume, and the voxel resolution on that would be about 150 to 200 microns. A nice thing about the system is we can also zoom in. So this is an example of a corn kernel. If we zoom in, and there's a software called SubPix, which gives us a little bit more resolution, we can get to about the 5 to 10 micron voxel size. So it's a very versatile instrument. And just as a real life example of this is a corn cob we can scan, and that's a kernel that came off of it. And so we can measure lots of stuff within this corn cob, and I don't know if you can see this, but also there's the little maize embryo. I think we're close to being able to count the number of leaves, things like that, endosperm. So a lot of information in this that we then have to extract using computation. Getting back to the roots, which is really the why we got the system in the first place, to look at the hidden half of plants. So this is a corn plant grown in a sort of a expanded clay, and what you're looking at is just a slice turned like this. And as the movie goes, you can see the stem and you can see the crown roots here, nice and round. As the movie goes, maybe you can track these, but after a little while, once they start to get small, you're probably having a hard time tracking those. So this is really the magic, because what we have to do is extract these roots back out of the reconstruction. So this is where root segmentation is an issue. So segmentation is just a general term for teaching a computer what's foreground and what's background, which objects of interest and what's not, so that you can then measure your object. So again, Ni is working here on automated or semi-automated methods for tracking the roots. And if you look carefully, there's roots that are missed. There's some things in here that are not. So in the end, we get a reconstruction, and it's pretty good. I mean, there's a lot of missing roots, and there's some stuff that's not roots, but we're getting there. This is an active area of research in my group, and just in general in the field of imaging. Sometimes segmentation's easy. Often it's quite hard. As Mike showed some examples of, there's certainly a lot of applications for machine learning and AI and this pattern recognition. So here's an example, again, the collaboration with Valent. It didn't do such a great job. This is a soybean, and you should be looking at, these things. We're looking for nodules. Those are definitely not nodules, or they're the world's biggest nodules. But you can actually see them if you look carefully here. But the point is, we're kind of getting there, but we're not quite there yet. But You know, again, as Mike mentioned, if you have very talented students in the lab who are studious and focused and methodical, you can clean these up with manual segmentation. So now this looks much more like a soybean root, and again, we can measure the litany of univariate traits, and we can also now identify the nodules, count the nodules, how big are the nodules, how are these changing over time. We can now capture that information. Okay, so that's the work we're doing there. Again, I'm just going to kind of go through stuff since this is a tech-forward kind of meeting, just to give you some examples of what we can see. So not just roots. This is a cassava root system. These are the storage roots and the fine roots, but not just the external morphology, also the internal morphology. So this actually is a little slice of the stem. It's just to show you that there's a lot of information here. For example, these are roots that are about to emerge through the stem. There's vascular tissue, things like this. We think we can possibly use this in disease progression. For example, to study cassava brown streak virus as it starts to develop inside of a tuber. So that's sort of an ongoing side project in the group. But the idea is it's not only that we can see outside of the plant, but also inside. So here's another example. This is a maize stalk. These are the crown roots, and one emerges with each leaf. So we can actually see inside the vascular connections between root and across the leaves here in the maize plant. So again, there's nothing quantitative about this. This now requires some person or people to come in and actually extract features from this, but the information is certainly there. And so X-rays can do a lot. Here's something that's a little bit easier to see. This is a developing tassel in a maize plant. So when we think about flowering time and environmental effects that affect flowering, we often think about it, well, when the tassel pops out. But actually, the primordia, right, when it's only even smaller than this, a few cells big, is more like the V4, V5 stage, right? It's inside the stem, and it's been growing there for a long time. So it may be that the relevant time point when we want to understand the environment is way back in time. But you can see just from this, I can predict how many leaves there are going to be on the plant before the tassel emerges, for example. That's sort of an obvious thing. I could probably count that by hand. And so this is an example of what it looks like a little bit older. So now we can see the individual florets. We can measure those traits, et cetera. Here's an example just for mutant analysis, right? So this might be an architecture that's kind of hard to capture. Like how would you measure it by hand? And Blake wasn't sure about this, but we can capture it inside of the stem as it's developing, and again use computation to measure how that's different from like a wild type. Here's another example in Ceteria inflorescence of just, so here's a mutant and here's the wild type. So that gives you an idea of some of the stuff we can do. Getting back to roots before I run out of time completely, one thing that we're doing is trying to extract more information out of the three-dimensional architecture. So I told you about dirt, but that's really 2D, so we're trying to extract information from something we've flattened down into 2D. We can scan these in about 2 or 3 minutes, depending on how big it is and what resolution. So we've been doing this by the hundreds. So now, if I want to quantify the differences between these two, I can use dirt, but I can also use three-dimensional data. So here's what the three-dimensional. It's very complex, and we're working with some topologists and computer graphics people to clean these up because there's actually a lot of loops and noise. But nonetheless, we can capture a lot of information from this. So I won't go through this experiment, but just to say that we basically took the NAM founders, which sort of encompass maize genetic diversity. We planted these out in our field in Columbia, Missouri last year, and we dug every week the roots up. So we have a time series, sort of a manual time series. But a subset of these at weeks 4, 7 in tasseling, we also scanned. So it's about 250 plants. I'm not showing you 250, only 3 that we've analyzed so far. So at tasseling, this is a principal component analysis just to show you for the 2D dirt traits, all the things we can measure with that 2D software. Principal component 1 does a pretty good job of separating Kentucky 21, these dots, from B73 and Missouri 17, but it can't really separate B73 and Missouri 17. When we take the X-ray data into account, all of a sudden we can now cluster B73, KY21, and Missouri 17 much better. So I think that's because the information content's higher. It's allowing us to separate these things. Discriminatory power is really what matters to us most. And if we really want to, we can go back and look at the trait loadings, and we can see that solidity, for example, is driving this and we can actually quantify each of the individual univariate traits as well. So we can get back to the biological insight. if we want to. But really what matters is discriminatory power. So each of these individual traits don't mean that much to me personally. It's just the ability to separate, and I think that will increase our ability to identify genes. So in the final bit of time, there's a section I won't get to, but just going back to this PET, this is sort of a really interesting technology. Again, you're looking at carbon moving. So we feed this plant radioactive carbon-11. It's a short-lived radioisotope, very energetic, and it's just fed. It takes it in, it does photosynthesis, and starts moving it down. That's an imaging artifact. So you're looking at real time at the carbon moving down. The reason this is so interesting is because now we're actually looking at physiological dynamics. Right? We're looking at what's happening inside the plant that's driving growth, and we're able to overlay that, it's multimodal imaging, with what's actually, with the morphology of the plant and how it's changing. So you saw this, that's Ty's plant PET system, and now we can look at not only carbon allocation and plant growth decisions over time as a function of development. So early on, there's no carbon going to the roots, and later we see that develop. These are 3 maize plants, or subjects as they're called at the med school. We can also look at this dynamically in real time. So we have about 2.5-hour window with carbon-11. I should mention Margaret Frank, who's about to start a faculty position here, has done a lot of work with nitrogen. There's plenty of other radioisotopes you can use as tracers. Carbon, just a really easy one. It's a gas. The plant takes it up and you can see it. But so here's, I don't have much more time, but here's a quick example again of some biological insight. So this is the optical gel system. This is a very young plant where only 1 leaf has emerged. And so you can see there's no carbon here in the roots. You see it in the shoot here. These are different bed times where the shoot's not being shown. The only carbon going to a root at this stage is this crown root here, so which is a shoot-derived root, and you can see a little spot there. The next day, when the next leaf emerged, all of a sudden the dam broke, the plumbing changed, the seed ceases to be the source of energy for the roots, and now the roots are growing, being fed from the leaves and the photosynthesis. So this is an interesting biological insight I think we can capture with this. It would have been hard to do otherwise. So ultimately, we're using optical methods, but now that we have the X-ray CT, or X-ray PET CT is actually where we're moving next. But the optical methods allow us to overlay these two and actually ask for a given amount of carbon at a root tip, how much should that root grow later? So this is something we're modeling, and we can see changes. For example, the jar we have to use is kind of small, so when the roots hit the side of the jar, we can actually see a difference in the relationship of growth to carbon allocated, so actual plant-environment interactions in real time. So if we wanted to for example, to query the transcriptome or the metabolome when 2 roots interact, if we wait till the roots have already grown, we've missed the dynamic event. The dynamic event that triggered the growth and was driving it happened before, and that's what we want to capture. So I won't have time to explain a mathematical technique that we're using similarly, but I'll show you some cool grape rachises. So we can use this mathematical method to capture comprehensive phenotypes. This is from the USDA collection. So Jason Londo, who's USDA and also here at Cornell. This represents grape genetic diversity, and just to show you with this mathematical method, we can actually discriminate different grape rachis architectures very well. We're hoping to use this now for root and shoot whole plant architectures. So you can read, we have a preprint here in bioarchives, work we've done in tomato, also a really nice essay in American Journal of Botany if you want to know more about mathematical approaches to, once we have the data, to actually capturing information in a comprehensive rather than ad hoc and discrete way. So I'll stop there. There's my group at the Danforth Center, several funders including the NSF, USDA, and Valent Biosciences. And if there's time, happy to take questions. Thank you.
One question.
Do you phenotype your real populations both in the gel and through shovelomics, and what are the correlations?
Yeah, so I skipped over that slide, but the Plant Genome Project, we're explicitly doing that, and we're not finished yet. But instead of correlating a trait we can get from a gel, which is very high resolution, and what we can get out of the field, which is sort of low resolution, we're just comparing at the genetic level. So do we find SNPs or QTL in common from gel from the field? And not only are we doing the shovelomics or the superficial roots, but actually mini-rhizotron imaging. That's what Andrew Leakey's doing. So are there any genetic signals in common, which I think is much more solid ground than traits, which vary depending on how you measure them, for example. So thanks. Yeah. Okay, thank you.
This has been a production of Cornell University, on the web at cornell.edu.